Every year, over 120 million rodents are used in biomedical research. But the data from these models is flawed. Why? Because when they interact with humans, their behavior and even their biochemistry can change. Studies have reported an altered blood glucose level and corticosteroid level. A study published in Nature even found that even the smell of a technician could lead to stress-induced pain inhibition, which distorts the results. We have a solution. It's an injectable, continuous monitoring device that can transmit the data without direct handling. It's minimally invasive, batteryless, wireless, and physically flexible enough. This means that our device is foldable, can be folded like this. Today, we can collect pH values and ECG, and we're planning to expand these functions to iron levels, blood pressures, and even brain waves. So picture this. It's a device that can be easily implanted. You just simply need to tap your mobile phone or transmitter to the animal housing to power this device and collect the data. There's no need to directly handle these small animals, therefore drastically reducing the stress and anxiety. This means that our scientists will have a more credible data and it's a better welfare for the animals. It's a win-win. Today, we've already achieved in vitro testing of our ECG functions. The next 2 stages we're planning to do in parallel are running an in vivo test and developing new functions. We're raising £1 million to complete these tasks. Please join us to refine our preclinical data. Thank you.
Well done, absolutely perfect time. Gents, you must have a couple of questions for this.
The obvious one, regulatory.
Yes.
So quite a tough regulatory environment. So have you spoken to the regulators and what's been their feedback?
But as for our device, the target is the research animals, so it's not classified as a medical device. So we do not need to have a clinical trials. Therefore, our device will be on market faster than like traditional medical device that will be used on humans. So for our device, if a research team want to use it, it will have to be approved by the home license, the home department, and the the ethical group that's different in different countries. So if your license include this, if you include this in this license, then you'll be fine to use it.
So I've got a more technical question. So you're putting these devices in animals. Once you've taken your measurements and get the device back out.
So the device is inside the mice, but the data is transferred by the transmitter. The transmitter is you can set the transmitter on the animal housing, and it will send the data from the device through the antenna to a mobile phone or a computer software.
My question was, how do you, once you put the antenna into the animal, how do you then remove the antenna once you've finished your experiment?
So hopefully the device is like injected to the subcutaneously. So the scar tissue won't be very strong. So after all the animals, let's say it's retired, you can just simply use a forceps to take it out like very easily.
And market adoption, just in terms of comparison to current gold standard in. So how do you compare to the current methodology, current gold standard in that?
Okay, so compared to the direct handling traditional method, our method will like definitely reduce the stress. And one of the competitors, like the AD instrument, they have implantable biosensors to they also monitor the ECGs. But what our device has the advantages is that our device is multimodal, so that we can have 2 functions or even 3 functions on one device. But their device like only have 1 function per device, so that's one of our advantage. The second one is that our device is batteryless, so it's powered by the transmitter. There's no need of battery inside the device. And lastly, our device is minimally invasive because it's injectable. It do not need surgery to implant it, and for a competitor, they need to have an easy surgery to implant the device.
Mentioned huge number of animals used in research across the world. What percentage do you think, or what proportion do you think will adopt your technology or convert?
So for now, our first target would be rodents, let's say rats and mice. We would believe it's account to around 20% to 30%, because the according to the specialists I've spoken to and the reports on the article, the most our device can be mainly used in neuroscience and natural behavior science. So in these two fields, scientists are more want to have the results for the animals in a more natural state to understand the mechanisms.
Well, thank you very much.